welcome back to your own channel Vedantu NEET English. This is Basavaraj, your biology master teacher. Students, this is a one-shot lecture on the topic mitosis from the chapter cell cycle and cell division. Now, students, this topic is very important along with meiosis because this particular topic, right, will help you to understand and make sure you entire concept of biology becomes much more clear. So, students, if I'm properly audible and visible to all of you, quickly add a fire emoji in the chat, students. If I'm properly audible and visible to all of you, students, quickly in the chat, all of you. If I'm properly audible and visible to all of you, can I get a fire emoji in the chat? Because let's just start, students. Let's just start. We cannot wait any further, right? We cannot wait any further. Now, students, in the previous class, we all of us studied, right? In the previous class, we learned that in a cell cycle, right, students, in a cell cycle, there is something called as an interphase. Interphase is the preparatory stage. In this preparatory stage, we have G1, S, G2, gap 1, S, gap 2, that is synthesis. Here in the S phase, the DNA replication happens there. Done. Now, today's class, we will be focusing on this entire M phase. In this entire M phase, students, we can see there is karyokinesis, cytokinesis. In the karyokinesis, there is a division of nuclei here. Here in the karyokinesis, we have in total of 1, 2, 3, 4. We have in total of 4 stages in the karyokinesis here, right? Prophase, metaphase, anaphase, telophase. 4 phases in the case of karyokinesis. Students, all of you, PMAT in the chat. Students, all of you, PMAT in the chat. All of you, every single one of you, students, every single one of you, PMAT in the chat. PMAT in the chat right now. Now that is karyokinesis, right? Then what is cytokinesis, students? Cytokinesis, cyto means cytoplasm. Cytokinesis is division of cytoplasm after karyokinesis. So in today's class, I will draw and I'll give you the notes right now itself. So I want all of you to open your book and start making the notes with me right now, students. So I want all of you to open your books and start making the notes right here, because I will make sure you understand, draw, and completely remember all the 4 stages. Okay, students, PMAT, all of you, PMAT. Now, here we can see, students, karyokinesis is prophase, metaphase, anaphase, telophase, PMAT. P-M-A-T. PMAT right here, students, PMAT. This is right here is PMAT. Prophase, metaphase, anaphase, telophase, that is called as PMAT. Okay. Now, students, now shall we start the class with prophase, students? Let's start the class with prophase. Okay. Now, before we go to prophase right now, all of us know there is something called as interphase. Yes or no, students? There is something called as interphase. Now, how do you think a particular cell looks like in interphase? Now, let's say this cell right now is in interphase right now. Let's say this cell right now is in interphase. Okay, interphase. Now, students, in interphase right now, do you think we find chromosomes? No. Chromosomes are not visible in the interphase. As a matter of fact, students, here I have the nucleus. Now, inside the nucleus, the entire DNA is present as thread-like structure. Can anyone tell me, students, what is this thread-like structure? Anyone in the chat? In the case of interphase, the entire genetic material, entire genetic material, right? Genetic material is present in the form of chromatin. Now, this is what is happening in the case of interphase. So please remember, students, in the case of interphase, this is how the DNA looks like, or genetic material looks like, genetic material. Now, in interphase, we have G1, S, and G2. G1, S, and G2. Now, after this, the next stage is going to be the entire prophase. Student. So this right here is the prophase. Okay, prophase. Now, what exactly happens in the case of prophase? Please understand and draw with me. Notes, Telegram channel, I'll give it to you, students. I've already given to you on human reproduction. I'll upload today itself. Entire chapter I'll give it to you together. Don't worry. Now this, let's say this is your prophase right now. What exactly happens in the case of prophase? Okay. Let's write the notes and then I'll teach you what exactly happens in prophase. Students, I want all of you to remember, prophase is actually the one of the longest phases in the karyokinesis, right? It is one of the longest phases, right? It is the one of the longest phases. Longest phase of karyokinesis. It is the longest phase of karyokinesis. Now what happens, students? Here there is an initiation. There is a initiation of condensation of chromosomal material. Now can anyone tell me what is this chromosomal material, students? This chromosomal material is undergoing condensation right now. It is undergoing complete compression. Now I'll give you a small visual representation. Students, do you know what is woolen sweater? Okay, you know, all of you know what is woolen. Now imagine there is a long woolen thread. Now this long woolen thread is like chromatin. That is your chromosomal material. That is nothing but the chromatin. Chromatin is the chromosomal material. Now if this long thread called as chromosomal material, if it undergoes condensation. Now students, all of you tell me, if you take the woolen and if you try to compress it, do you get a ball? All of you in the chat. So if you take a woolen thread, and you undergoes complete, like if you keep on rolling it, if you keep on rolling it, right, it becomes a ball. Now that is called as condensation of your woolen. Now your woolen is like chromatin. Now if you take the chromatin and you compress it, we get woolen ball. In this case, if you take chromatin, if you condense it, we get chromosomes. Okay, students, I've told you this before also, right? Multiple times I've told you, right? If you take a chromatin, long chromatin here, if you take, and if you completely compress it, this chromatin will have a shape. This chromatin will get a shape like this, something like this. Will get a shape like this, students, shape like this. Now this right here is called as chromosome. So that's how you get chromosome, right? You take a chromatin, you condense it, you get a chromosome. Now that is what is happening in the case of your prophase. In the case of prophase, there is initiation of condensation of chromosomal material. The chromosomal material undergoes condensation. Simple as that, students, simple as that. Now this entire prophase can be divided into two parts. Okay, we have early prophase and we have late prophase. Okay, we have early prophase and late prophase. Now understand what exactly happens in early and late prophase right now. In the case of early prophase, what is happening, students? In early prophase, there is condensation of chromosome. Yes, we can get chromosomes right now. Chromosomes, we are finding chromosomes. Apart from condensation of chromosome, there will be something called as movement of asters or movement of centrosomes. Actually, there is something called as movement of centrosome. Now students, if I am mentioning centrosome, am I talking about your plant cell or animal cell? Anyone in the chat who can tell me? If I am mentioning movement of centrosome, all of you should know one centrosome has two centrioles. Right? One centrosome has two centrioles. So these centrosomes, which are present only in the case of animal cell, will start move towards the opposite poles. Let me show you. Here, let's take a nucleus, students. Let's take a nucleus here. Here we have the nucleus. Okay, let's say we have the nucleus here. In this, inside the nucleus, all of this genetic material, the chromatin, undergoes condensation to look something like this. They'll start looking something like this. They'll start looking something like this here. Inside the nucleus. Inside the nucleus, the chromosomes are getting condensed right now, condensed. Now, apart from the students, apart from that, there will be movement of centrioles or centrosome altogether. So the centrosome, it is present somewhere in the cytoplasm. In the cytoplasm, it will now move towards the opposite poles like this. Here we have the centrosome, which has a layer around it called as amorphous pericentriolar space. Amorphous pericentriolar space. Now this right here is called as centrosome. There are two centrosomes here. Students, if there are two centrosomes, can anyone tell me how many centrioles do I have? I have two centrosomes and four centrioles. I have four centrioles right now. They're moving towards the opposite poles. Now what happens, students? Now what happens? From the centrosome, there will be a release of microtubule. All of you remember, microtubules are basically a type of cytoskeleton here. And the microtubule I am talking right now is called as spindle fiber and asters. So from here, there will be spindle fiber release. Spindle fiber is being released from here. Spindle fiber released from here. And at the same time, students, here there will be aster release. Asters on the lower side. Asters on the lower side. Now you'll ask me, sir, what is the function of asters? Why do we need asters? Please understand, students, please understand. Let's go back here. Please, let's just go back here and write down. So I told you there is condensation of chromosome is happening. Movement of centrioles, centrosome is happening towards the opposite poles. Now here, there are two types of microtubules. There are two types of microtubules here. Okay. There are, in this, there are, let me just draw a box here. There are two types of microtubules here. One is called as, students, there are two types of microtubules here. One is called as your spindle fiber. Spindle fiber, right? One is called as your spindle fibers. One more is called as asters. Is called as asters here. Now, can all of you see, students, here? Here we have the spindle fibers here. One second, students. Here we have the spindle fibers right here. Can you see? Spindle fiber. And this right here is called as aster. Aster here. Now, students, spindle fiber will help in what? Spindle fiber will help in attachment to the centromere. Now, this spindle fiber will attach to centromere. Centromere of chromosome. It will attach to the centromere of chromosome. Now, this aster here, it will help in anchorage. It will help in anchorage. It will help in anchorage. It will make sure the centrosome doesn't go anywhere. This aster will anchor down, make sure the centrosome doesn't move anywhere. Now, students, this together, spindle fiber plus aster, this spindle fiber plus aster is called as what? Together, they are called as mitotic apparatus. Mitotic apparatus. See, students, can all of you see here? Here we can see the prophase. Initiation of condensation of chromosome is happening here. chromatin condensation is happening here. It is condensation is happening here. Then there is movement of centrosomes. Centrosome has two types of microtubules, right? Right. Centrosome. Actually, kinetochore. Exactly. Technically kinetochore only. Technically kinetochore itself. Technically kinetochore itself. This spindle fiber is arising from this amorphous pericentriolar space. Then there is asters here. Together they are called as mitotic apparatus. They help in anchorage, students. They help in anchorage. Now, students, remember this particular structure, aster, is only found in animal cell. Aster is found only in the case of your animal cell. Asters are not found, students. Asters are not found in the case of your plant cell. Please remember, students. Please remember that. Now, this is what is happening in the case of prophase. Can you see here, students? So this is your early prophase. There is spindle fiber plus aster. Together called as mitotic apparatus. Together they are called as what? Mitotic apparatus here. Mitotic apparatus. Okay. Now, what exactly happens in the case of late prophase? Understand, students. In the case of late prophase. In the case of late prophase, students, there will be something called as disappearing. The entire Golgi complex, endoplasmic reticulum, nucleolus, nuclear envelope will start to disappear. Disappearing of organelles like Golgi complex, ER, right? Different organelles, nuclear envelope, everything will start to disappear right now. So this is how it looks. By the end of it, you can see this entire nuclear envelope will start to have certain spots in the middle. This will somewhat start to look like this. It will start to disappear. Okay, it will start to disappear right now. That is happening in the case of prophase. Now, students, prophase with that is over. So I'll give you a complete recap of prophase. Listen to me very carefully. So here we have interphase. Genetic material is as a chromatin here in this case. Completely thread-like structures. Then what happens? There is chromatin condensation is happening here. Entire chromatin is undergoing condensation. Then there is a movement of centrosomes. One centrosome has two centrioles. It is covered by amorphous pericentriolar space there. Now from this centrosomes, there are two types of microtubules. These are nothing but microtubules. Two types of microtubules. One is called as spindle fiber. One is called as aster. Spindle fiber as well as aster release is happening. Here, spindle fiber will have basically attached to the kinetochore of chromosome, and this aster will help in anchorage here. Aster is only found in the case of animal cell. Spindle fiber plus aster is called as mitotic apparatus. All of this is happening in the early prophase. Okay. Now, as we come to late prophase, disappearing of organelles happening, Golgi complex, ER, nuclear envelope, everything is disappearing by the end of it. Now, with that, a cell is ready now to go to the next stage, that is called as metaphase. All of you students. And students, can you see here? This is the entire centrosome. Centrosome has two centrioles here, which are perpendicular to each other. And here we can see the asters here. Aster here. Here we can see the spindle fiber. Spindle fiber here. Spindle fiber is here. Now they're attaching to the chromosome right now. Attaching to the chromosome. Okay. So this is what is happening. Initial chromatin condensation. It becomes a ball here. And then we have chromosome formation. So by the end of prophase, we can see the chromosomes inside a cell. Chromosomes becomes more and more visible. Okay. Become more and more visible. Okay. Students, any doubt till now? Students, any doubt till now? Quickly, all of you. All of you. Students, any doubt till now? Prophase done? Okay, now students, now you can see NCI diagram, early prophase, late prophase. You can see condensation here, students. You can see condensation has happened here. Condensation has happened here. Okay. Now, students, now what exactly happens in the case of metaphase? Let us go back to this diagram. Okay. Now let's draw a diagram from here itself. It'll be easy to all for you. Now what happens? Understand, students. Now let's draw one more cell here. Now this right here is metaphase. This right here is metaphase. Understand. Now in the case of metaphase, what is happening? In the case of metaphase, now all the chromosomes will start moving towards the center. All the chromosomes, students, all the chromosomes will start to move towards the center right now. Like this. Here we have the chromosomes. All the chromosomes will now start to move towards the center. Like this. And in the corners, what do we have here? Here we have the centrosomes in the corner. From here, we can see the spindle fibers rising. Some spindle fibers do not attach, they just pass through. Now, this is what is called as metaphase, students. This is what is called as metaphase. Okay. Now, this is called as metaphase right now, students. Okay. Now, here chromosomes become fully condensed in metaphase. And that is why they say this is the best stage to study the chromosome morphology also. Okay. And students, you can see here this middle part here. Now, all of these are chromosomes here. All of them are aligned at this part here, right? This part here. This right is called, this middle part is called as metaphasic plate. Metaphasic plate, also called as equatorial plate. Equatorial plate. Here. Now, I want all of you to understand in metaphase what exactly is happening, students. Exactly is happening, students. Darshini, yes, I will reply. Don't worry. I'll reply, students. I'll reply. Now, I want all of you to understand. Let me just draw one more diagram here for your convenience. Right? You get a better perspective. Here was this is the metaphase one, right? Now, here what is happening is, please focus very carefully right now. Very carefully. Now, can all of you see? I'll just draw one chromosome and I'll show you what exactly is happening here. Okay. Okay. This is one chromosome right here. This chromosome middle part is here. Now, let's focus this one vertically right now. Okay. Same thing I'm drawing right now, students. Same thing I'm drawing right now here. Okay. Now, this center part is called as centromere. This center part is called as centromere here. Centromere. These are called as sister chromatids. Now, around the centromere, students, around the centromere, what happens, students? Around the centromere, what is happening here? If all of you properly notice, around the centromere, there is a cup-shaped structure here. There is a cup-shaped structure here. Here also we can see. We can see here also a cup-shaped structure. Now, this cup-shaped structure is called as kinetochore. It is called as kinetochore. Now students, I want all of you to understand the spindle fibers are attaching always to the kinetochore. The spindle fibers are attaching from here. If they come right here, let's just, I'll draw only one side, students. Here we have the centrosome right now here. From here we have the spindle fibers arising. They will always attach to the kinetochore, students. So all the spindle fibers will always attach to the kinetochore. Very important line, students. Very important line. This happens in the case of metaphase itself. Okay. So first of all, what happens here, students? First of all, what happens? There is something called as movement of chromosome to the center is happening here. This has a name, students. It is called as congregation also. It is called as congregation. The movement of chromosome to the center here, right in the center, is called as congregation. So first of all, what is happening, students? There is something called as congregation is happening here. Okay. Okay. Sir, in prophase or in metaphase it attaches. Govirshan, it happens in metaphase. Attachment to this attachment of spindle fiber to the kinetochore. It is happening only in the case of metaphase starting itself. Metaphase. Spindle fiber formation is happening in prophase, but attachment is not happening. Attachment only happens in the case of the metaphase right now. Here there is a formation of equatorial plate here. There is formation of equatorial plate or metaphasic plate, whatever you call it. Okay, metaphase. Now here, students, the chromosomes are fully condensed right now. Chromosomes are fully condensed. Chromosomes are, let me just write it in some other. Chromosomes are condensed right now, fully condensed right now. So they say this is the best stage to study chromosome morphology, right? Chromosome morphology, best stage to study the chromosome morphology, students. Here now, students, can anyone tell me if this is a diploid cell? If this is a diploid cell right now, in this diploid cell, how many chromosomes do I have? Anyone? In this diploid cell right here, students, how many chromosomes do I have here? Chromosomes. Anyone? So this is a diploid cell right now. Diploid cell will have 46 chromosomes right now, students. It will have 46 chromosomes. And every single chromosome has 2 chromatids. So can I tell there are 92 chromatids here? All of you? There are 46 chromosomes and 92, all of you. What is 46 into 2? Anyone who can tell me? Students, what is 46 into 2? Anyone? What is 46 into 2? Anyone, students? What is 46 into 2? It is happening in the case of metaphase. All of it tell me in the chat quickly. All of it tell me in the chat quickly. What is 46 into 2? All of you. And you can see, students, here at the equatorial plate in metaphase of mitosis, there are 2 sister chromatids. There are always 2 sister chromatids arranged here. 2 sister chromatids are arranged here. Okay. 2 sister chromatids are here. Okay. Best stage to study chromosome morphology because chromosomes are fully condensed right now. Okay. Again, please remember main point here in metaphase. Spindle fibers are attached to the kinetochore. This is very, very important. This is very, very important. This is very important line. It is a very important line, student. This is a very important line. Okay, very important line right now, students. Spindle fibers attached to kinetochore. Now, this is what is happening in the case of your metaphase. So, movement of chromosomes to the center, they move towards center. That is called as congregation. There is a formation of equatorial plate, also called as metaphasic plate, as we can see here. Here, equatorial plate or metaphasic plate here, right in the middle right now, students here. Okay. Now, apart from the students, what happens here? All the spindle fiber attached to the kinetochore, chromosomes are fully condensed. Best stage study the chromosome morphology. With that, it is over, students. With that, it is over. Okay. With that, your entire metaphase is also over. Okay, metaphase is also over. Now, students, you can see here, with the help of this something called as chirogram. With the help of chirogram, they find diseases, they find any chromosomal aberrations, chromosomal changes with the help of chirogram. And every single chromosome here in chirogram, it is present at metaphase actually, because morphology is clearly visible right now. So chirogram is used to find chromosomal abnormalities, and it is best studied in metaphase. Okay, best studied in metaphase, students. Okay, now students, now we come to anaphase, the most important. And if you understand anaphase, the entire concept is clear to all of you. So please follow very carefully for anaphase what exactly happens in anaphase. Let's just go back to this particular slide here. Okay, now let me just draw one anaphase here. What exactly happens in the case of anaphase? In the case of anaphase, what is happening, students? In the case of anaphase, please understand, students, if this right here, in the case of anaphase, what is happening is, if this is the chromosome right now, here we have the centromere. Here we have all the chromosomes right now. In the case of anaphase, what happens is that here we have the kinetochore. Here we have the spindle fibers, and here we have the nuke centrose. Right now, students, just try to imagine if I hold this part and I hold this part on top. Now, what if I start pulling out from the both the sides? What will happen? Think visually. Try to understand. I will hold on the both the sides and I'll just pull it apart. What do you think will happen? Students, let me just draw the vertical section for you. Right? Vertical section for you here. All of you understand better. Can I tell this is your chromosome right now? Can I tell, students? Can I tell? Here we have the kinetochore. Here we have the spindle fibers. Okay. Now, if I hold from both the sides and I pull it apart, will this become two pieces? Will it become two pieces right now? All of you in the chat. Yes or no? This will completely split in half. This will completely split in half. And once it splits in half, what do we get? If it splits in half, we end up getting a structure like this. Yes or no? We end up getting a structure like this. Here we have the kinetochore. Because we pulled it from both the sides, it broke open. What broke actually, students? There is something called as the splitting of centromere is happening here. Actually, what is happening, students, here? There is something called as splitting of centromere. Now, if centromere splits, what happens? There is separation of, these are called, students, what? These are called as sister chromatids. Yes or no? Sister chromatids. There is separation of sister chromatids. So these sister chromatids will separate open right now, separate open. Now, students, this is how the actual anaphase looks like right now, students. Anaphase. Okay, let me show you. Here we have the centrosome with amorphous pericentriolar space, and here we have the spindle fibers. Spindle fibers. At the end of spindle fiber, students, at the end of spindle fiber, we have the centromere. Centromere is right here, and we have the chromatid like this after splitting. So this is how it looks after splitting in the case of anaphase, students. Okay. Students, is my voice breaking? All of you in the chat quickly. Is my voice breaking for all of you? No, right? So there was a centromere right in the middle, right, students? Here. There was centromere here. This entire centromere split open. And always remember, students, always remember, the centromere will be first, centromere will be facing towards. Centromere will always be facing. Centromere will always be facing towards the centrosome. Centromere towards the centrosome, and the chromatids will be trailing behind. Here, students, understand, these chromatids will be trailing behind. It is a line of NCERT, students. It is a line of NCERT also. Okay, it is a line of NCERT also. So all of these chromatids will be trailing behind. Now a question to all of you, students. Now what is happening is that in the case of anaphase, students, can anyone tell me how many chromosomes were there in the starting? In the starting, there was 23 pair of chromosomes. Yes or no, students? In the starting, there was 23 pair of chromosomes. Yes or no? There was 23 pair of chromosomes. That is basically 46 chromosome in the start. Okay. Now I want all of you to tell me, students, is this a chromosome? All of you. Is this a chromosome? Yes. Is a chromosome? It is a chromosome. Is this a chromosome? Students, is this a chromosome? Yes. So technically speaking, both are chromosomes right now. How do you identify, students? You can easily identify that with the help of looking at the centromere. If you look at the centromere right now, one centromere, one chromosome. One centromere, one chromosome. But technically speaking right now, this one chromosome has two chromatids. This one chromosome has a single chromatid here. Now, in this anaphase, what happens, students? In the case of anaphase, every single daughter chromatid will act like daughter chromosome. So temporarily, students, what happens? Temporarily, very important line, students, very, very important line. Okay. Temporarily, what is happening here? Temporarily, every single daughter chromatid is acting like a daughter chromosome. Obviously, students, because these chromatids will become a chromosome later on. Yes or no? Now, students, can anyone tell me, technically speaking, did we have? Technically speaking, okay, students, technically speaking, we had 46 chromosomes right now, and we had 92 chromatids here, because every single chromosome in the starting, every single chromosome had 2 chromatids. We had 92 chromatids here. Now I am telling you, every single daughter chromatid right now, after splitting, every single daughter chromatid is a chromosome. So can anyone tell me right now, students, how many chromosomes do I have right now temporarily in anaphase? Anyone, students? Anyone? All of you? Anyone? See, I told you in the starting we have 46 chromosomes right now here. We have 46 chromosomes here. And I am telling you, we had 92 chromatids here. Now, every single chromatid here, all the 92 chromatids are temporarily behaving like a chromosome. So, can I tell, students, right now, temporarily in anaphase, we have 92 chromosomes. 92 chromosomes. So, students, if n, right, if n is 23, 2n is 46, 3n is 69, 4n is what? Anyone, students? Anyone? 4n is 92. Okay. All of you. 4n is 92. So temporarily, this particular anaphase one, not this one, after splitting, right? After splitting this one. Where did we go? Here, temporarily, this is a 4n condition. Yes or no, all of you students? Yes? All of you. This right now is temporarily 4n condition right now. 4n condition. Now that is what is happening in the case of anaphase, students. That is what is happening in the case of anaphase after the chromatids separate out. Now, students, I want all of you to understand, in this particular anaphase, right, after the splitting here, this is the best stage to study chromosome shape. Actually, it is the best stage to study the chromosome shape. It is a best stage to study chromosome shape. Best stage to study the chromosome shape here. To study the chromosome shape. Let me just write somewhere up top, students. Right here. Best stage to study chromosome shape. Now, what do I mean by chromosome, students? Chromosome shape. Do all of you remember V shape, I shape, J shape? Students, do all of you remember the L shape, metacentric, submetacentric, telocentric? Students, do all of you remember? All of you remember, students? All of you, do you remember? Submetacentric, telocentric, acrocentric, all that can be studied at anaphase. All that can be studied at anaphase right now, students. Anaphase. Okay. So in anaphase, splitting of centromere is happening. Separation of sister chromatid is happening. Here, daughter chromatid is acting like a daughter chromosome. So temporarily in the anaphase, we have 92 chromosomes right now, students. 92 chromosomes. Okay. See, this is how it looks, right? It is trailing behind. See, the centromere is towards the pole and the two chromatids are trailing behind. Okay. Now, students, I want all of you to understand what exactly happens in telophase. Very simple, students, very simple. Telophase is the complete opposite of your prophase, actually. Complete opposite of your prophase here. So what is happening here, students? Let me just draw the diagram. We'll go back to that slide only because easy to remember, easy to tell all of you here itself. Okay. Now, what exactly happening in the case of telophase? in the case of telophase. In the case of telophase, students, what is happening is that the chromosomes all will appear, all will go to the opposite poles. All the chromosomes will go towards the opposite poles, like this. They'll go towards the opposite poles, like this. Some of the in later stage, they will again start, some of them will start getting turned into chromatin also, back to chromatin. Okay, and we can see nuclear envelope is appearing. Nuclear envelope is appearing also here. Okay, nuclear envelope is also appearing right now, students. Okay, so what is happening right now in telophase, students? In the case of telophase, right? In the case of telophase, students, chromosome cluster at opposite poles. Chromosome cluster at opposite poles, right? Opposite poles. They're clustering at opposite poles here, this here and here, right? Here, all the chromosome will start getting converted back to chromatin right now, to chromatin. Now, in this process, they will lose their identity. They will lose their identity because the entire shape is going away now. They're becoming chromatin, again back to chromatin. So they're losing their identity right now. Losing their identity right now, students. Okay, and everything is reappearing. Reappearing is happening. That could be the Golgi complex, endoplasmic reticulum, right? Nuclear envelope. Everything is reappearing, students, reappearing. That is what happens in the case of telophase. Telophase, students, it is reappearing. Now I want all of you to understand, students. I will show you one entire animation to get the entire flow set once. Okay, so let me just go from starting once to the end. What did we learn today? What did we learn, students? We learned today. Okay, we learned that there is something called as interphase, M-phase, Karyokinesis, Cytokinesis, Prophase, Metaphase, Anaphase, Telophase. Cytokinesis, I'll teach you this one till now, it's pending. Okay, now initially what happens, we have chromatin, it undergoes condensation to become chromosome, and it is only visible in the M phase. It is only visible in the M phase. The chromosomes are visible only in the M phase here. Okay, now first we have the interphase right now, G1S, G2. Here we can see genetic material as a chromatin right now. Then what happens? Chromatin condensation happens here, initial condensation, and in the early prophase there is movement of centrioles or centrosome, actually the centrosome. Then there is a two types of microtubules formation here. One is called as spindle fiber, one is called as aster here. Aster help in anchorage, spindle fiber attached to the kinetochore here. Aster is found only in the animal cell. Spindle fiber is found both in plant cell and animal cell, both it is found, students. Okay, it is found in spindle fiber is found in both plant cell and animal cell. Okay, now together they are called as mitotic apparatus. Now in metaphase, what happens, students? Entire chromosomes are at the center right now. The center part is called as metaphasic plate or equatorial plate. In this phase, chromosomes are completely condensed and all the spindle fiber is attaching to the kinetochore. Okay, then in anaphase, what happens, students? The centromere splits, separation of sister chromatid is happening here. Here centromere will be towards the poles here, poles. Then we have telophase. Again, they were losing their identity here. They are becoming again decondensed, right? Nuclear envelope is reappearing. Everything is reappearing right now, students. Okay, notes are written here also today. See, early prophase, late prophase, prophase, what happens here? Okay, metaphase, movement of chromosomes to center, that's called as congregation, formation of equatorial plate, attachment of spindle fibers to the kinetochore. Best study to study the chromosome morphology using chirotyping, students. It is using chirotyping. Okay, anaphase, splitting of centromere, separation of cystic chromatids, starting from 23 pair, 40 chromosomes. We are ending with what? We are ending with 92 chromosomes right now, students. We are ending with 92 chromosomes right now. Okay, now students, then we have telophase, chromosomes clustered at the opposite poles like this. Chromosomes again go back to chromatin. In that process, they will lose the identity. Reappearing of Golgi complex, ER, nuclear envelope, everything starts to reappear. That is what is happening, students. That is what is happening at the entire thing. Okay. Now, students, let me just show you one small animation here. Students, see this. First prophase we'll see, students. First prophase we'll see. Can you see? Condensation is done. Movement of all of these are called as what? Centrosome, has two centrioles. Here we can see spindle fibers. Here we can see asters. They're going towards the corner here. Okay. Now, in the metaphase, what happens? Everything comes towards the center. That is called as metaphasic plate. Right in the middle, metaphasic or equatorial plate here. Now, in the case of anaphase, what happens, students? In anaphase, pulled apart. Splitting of centromere is happening, students. In anaphase, there is something called as splitting of centromere is happening, and they're pulled towards the opposite poles. Centromere is towards the pole, remember that. And chromatids are trailing behind. Okay. In telophase, what happens? They will start to cluster towards the poles here. Then there is a contractile ring formation here. Now this contractile ring will separate out when we come to cytokinesis. Students, cytokinesis. Okay. Just close this. Now, students, any doubt till now? Anyone, students? Any doubt till now? Students, any doubt till now? Okay. Now, students, we come to cytokinesis right now. What is cytokinesis? Cytokinesis is nothing but cyto means cytoplasm. Kinesis, division. Division of cytoplasm is happening, students. Division of cytoplasm is happening here. Okay. Now, in the case of plant cell, animal cell, it is very different. Very, very different. Now, understand what exactly happens in the case of animal cell. Okay. So, let's say this is an animal cell right now. Animal cell right now. Okay. Here we have one nucleus. Here we have one more nucleus here. Right. Here inside we have the chromatids with one chromosome. Sorry, one chromatid. Chromosome with one chromatid. Okay. Or most of them are back to chromatin. Most of them are back to chromatin stage also. Many are back to chromatin, thread-like structure. Now what happens, students? Here there will be something called as furrow formation here. That is called as furrow formation. Okay, furrow formation right here. Now furrow formation is because of phragmoplast, actually phragmoplast, right? Now students, try to imagine, I will take this and I'll try pinching it from the outside. From outside, I'm trying to pinch it from outside. Okay, so what will happen? What will happen, students? Okay, do you think it will become something like this right now? Yes or no? Pinching it more? All of you in the chat. If I keep on pinching it, it will start to become very, very thin. Very, very thin. Okay. Now, if I pinch it even more from outside, will it cut and divide into two cells? Yes or no, students? All of you. That's how we get two different cells here. And filled with chromatin. Filled with chromatin on the inside. Chromatin on the inside. And if you keep pinching it, this is what happens. This is what happens if you keep pinching it. Now, this is called as furrow formation. This is called as your furrow formation, students. Furrow formation. And students, remember, this furrow formation is happening from the outside. That is called as centrifugal. It's called as centrifugal. From outside it's happening, centrifugal. Now, students, understand what is happening in the case of your plant cell. Now, in the plant cell, we cannot pinch from outside. Not possible. Students, all of you, please smash the like button if you want the notes. Okay. Now, students, in the case of plant cell, what happens? You cannot pinch from outside. Not possible from outside. So, in the case of plant cell, what do we do? Students, all of you know, in your house, if your mother, after some time, how much ever she hits you, do you think it matters? Like, mummy, give me one shot, she'll be quiet. Now, when mother knows whatever amount of physical abuse is not possible, she cannot hit you anymore. What do you think mother use, students? Mother will use emotional blackmail. She will try to attack you from the inside right now. Yes or no? Same thing is happening here also. Now if I cannot attack it from the outside, what do? I attack from the inside right now, students, inside. So inside there will be something called as cell plate formation, and once the cell plate formation happens, it will start to segregate outwards this way. Let me show you, student. Let me show you. Here, student, see. This is called as furrow formation. Can you see? Actin filaments are there, phragmoplast from your Golgi complex. It will start to make furrow formation from outside. Can you see? First you see this. Slowly, the cell will divide into two. This is happening in the case of animal cell. Now, in the case of plant cell, can you see, students? There is something called a cell plate formation. Okay, right in the middle, there is a cell plate formation right in the middle, and then it will separate out. Now, that is what is happening in the case of plant cell. In the case of plant cell, there is cell plate formation, and they will move apart. Okay, that is what is happening in the case of your plant cell. Okay, so plant cell happens from the inwards. Now that is called a centripetal. That is called as centripetal type of separation from inside. This is centrifugal, from outside it's happening, outside here. That is called as centrifugal. Now that is called as your cytokinesis in the case of animal cell and plant cell. Now what is the significance? Growth and development, reproduction in unicellular organisms, repair and maintenance, growth in meristem, restoring nucleocytoplasmic ratio. All it is happening because of this. Now what is this, students? Restoring nucleocytoplasmic ratio. Imagine, students, if there is no cytokinesis, the cell is growing. The cell is growing. And if there is no nucleus dividing, will there be a difference between the nucleus and the cytoplasm? Yes, so students, imagine the cell is growing bigger and bigger because of cell growth. Cell growth is continuous and the nucleus is not dividing. So there will be difference in ratio between nucleus and the cytoplasm. We have to maintain that. So maintaining of nucleocytoplasmic ratio is done by mitosis. Students. Now here's the question to all of you, students. Here's the question to all of you. Who can tell the answer? Nandu, happy birthday, Nandu. Happy birthday. Students, quickly smash the like button, all of you. Quickly smash the like button. Students, where is the answer? Students, you cannot find centriole. You cannot find centriole at all. Centriole, as a matter of fact, is completely absent in the case of plant cell. Now, if there is no centriole in the case of plant cell, how do you think the spindle fibers are formed? Anyone? Think hard. If there are no centrioles, right, how do you think the spindle fiber is formed? Anyone? Anyone? How are they formed? How is it possible? Students, understand. In the case of your plants, they have something called as MTOC. They have something called as Microtubule Organizing Center, right? MTOC, Microtubule Organizing Center. Now, this Microtubule Organizing Center can serve as an organized assembly the microtubules required for spindle fiber formation. So they have something called as MTOC. MTOC will help to form the MTOC will help in formation of spindle fibers. Okay, MTOC, Microtubule Organizing Center. Okay, students. Now I want all of you to understand one extra concept here. This entire process of cell cycle, right? It is called as cyclin and CDK mediated. There is something called as cyclins. They are type of your proteins, and there is CDKs. That is cyclic dependent kinase. That is the enzyme. So this enzyme and this protein, when they join together, they become activated. They will completely control the entire cell cycle. Every single step of cell cycle is controlled by these two, actually, by these two. You can see, students, here. We have CDK4/6, CDK2, CDK1, CDK1, right? We have cyclins D, cyclin EA, cyclin A, cyclin B. So this CDK partner and cyclin together, they will control all the functions: initiation of DNA replication in S phase, preparation of cell for mitosis, triggering the mitotic division. So every single step in the cell cycle is controlled by cyclins and CDKs, cyclin dependent kinase, that is the enzyme. Okay, those two completely control the entire cycle. Now students, there is one more concept which I always teach, called as right, there is a checkpoint. Okay, there is something called as spindle assembly checkpoint here, right? Now there is something called as, let me just read for you. If spindle fibers are not attached properly, where during your metaphase, right, during your metaphase, if they are not attached properly, the cycle is paused until all the chromatids are properly attached to a spindle fiber. So sometimes what happens in nature is that spindle fibers do not attach to the kinetochore very well. So until the attachment of spindle fiber happens, right, the cycle will not go further. Students, try to imagine a condition where a cell is dividing into two half, but the chromosome number goes up and down. That can cause mutation, yes or no? That can cause a lot of changes in the body. It can cause. So always when a cell is dividing, it will make sure there is a checkpoint. After metaphase, it will make sure all the centromeres, all the kinetochore is attached to spindle fiber. Okay, a protein called as APC. Okay, anaphase promoting complex help the cell to proceed to anaphase. So unless we get approval, unless something happens, the cycle is paused, APC will come and it will push it to the anaphase right now. Okay, so please remember, this was a NEET PYQ, not given NCRT, but it is a NEET PYQ for all of you. Anaphase promoting complex help the cell to proceed to anaphase. So it will help the cell to get into anaphase right now. It's a NEET PYQ, APC. Okay, students. Now, all of you, what do you think will happen? What do you think will happen, students, if the cell is doing only karyokinesis but not cytokinesis? The nucleus is dividing, students. The nucleus is continuously dividing. Nucleus dividing. But the cytoplasm is not dividing only. Cytoplasm is just chilling there. What do you think will happen, students? What do you think will happen? Anyone? If there is karyokinesis, nucleus is dividing, but cytoplasm doesn't divide. Just try to imagine, this is one cell right now, right? This is one cell right now. One cell right now here. Now let's say we have two nucleus in the starting. Okay. Starting we have the two nucleus. Okay. Now these two nucleus will divide. These two will also divide. So we end up getting two more nucleus. Two more nucleus. Now these two nucleus will also divide into two more. They keep on dividing now. They keep on dividing. Students, by the end of, if this keeps on happening, what do we get, students? If this keeps on happening, can I tell we will end up with a multicellular structure? Yes or no? We might end up with a multinuclear structure. We might end up with a multinucleated structure. Yes or no? Multiple nucleus. Yes. So when there are multiple nucleus, right? When there are multiple nucleus like this, right? In one cell, that is called as syncytium formation. Okay. So that is called as syncytium formation happens. So karyokinesis is not followed by cytokinesis. That leads to multinuclear condition. That is called as syncytium. One of the examples is your liquid endosperm of coconut. Okay. Exactly, student. Telegram memes only. It's from my Telegram memes, right? Students, I share memes in my Telegram. Students. So this is what we get, a multinuclear condition called as syncytium. We get syncytium right now, students. Syncytium. Okay. Now we come to the last part of this chapter. Okay. To make the entire concept much more solidifying, I will teach you right now something called as a DNA content as well as chromosome number. Okay. Just try to follow with me step by step what exactly happens inside a cell. Okay. All of you students, all of you. Just try to follow through, students. Now first, what do we have? G1. In G1, this is how the chromosome looks like. One chromatid one. Here we have DNA content is 2,1, 2C in the starting. Now, in the S phase, there is DNA replication. So we end up getting 2 and 4C here. 2 and 4C. 2 and 4C. In G2, there is no change. In G2, there is no change here. We end up getting 2 and 4C. In the case of your prophase, same thing is there. No change in prophase also. Prophase, metaphase also no change. Metaphase 2 and 4C condition only. But as we come to anaphase, students, as we come to anaphase, what happens? Right, as we come to anaphase, this particular thing will split. Do all of you remember in anaphase? So we end up getting 2 chromatids. Yes or no, students? Do all of you remember? Do all of you remember, students? And I told you, students, here if you come back to anaphase right now here, if you go back to anaphase, didn't I tell you, students, temporarily it is in foreign condition because of 92 chromosomes right now. It has 92 chromosomes. So temporarily it is in foreign condition. It is in foreign condition right now. So we have foreign 4C condition, anaphase. Now what exactly happens in telophase? In telophase, students. Now here you will understand per cell what happens and per nuclei what happens, because this nuclei will become a cell later on, right? One entire cell will become nuclei, will become a cell. So let's understand, let's go back to telophase and understand, students. Let's go back to telophase here. Okay, let's go back to telophase right now, students. Where is our telophase? Here. Now in this telophase right now, in this entire telophase, we have 92 chromosomes. All these chromosomes have one chromatid. It has one chromatid right now. All the 92 chromosomes have one chromatid right now. Temporarily, this entire cell is in foreign condition, 4C condition, 4 and 4C. Now students, what if I tell you, out of this 92 chromosomes of one chromatid, half is going this side, half is going this side. So can I tell, at this point here, in this nuclei right now, we have 46 chromosomes. And in this nuclei, I have 46 chromosome. Again, one chromatid. One chromatid. Again, one chromatid. One chromatid. Again, one chromatid itself, students. One chromatid. Now I want all of you to focus. So out of this 4 and 4C, this entire cell, this one nuclei itself, can I tell is 2N because half came here. Can I tell this one is nuclei is also 2N right now? Out of this 4C DNA content, 2C went this side and 2C went this side. So when you ask me at telophase, right now, if you go back here, at telophase right now, per cell, it is 4 and 4C. But if you ask me per nuclei, it is 2 and 2C. 2 and 2C. And how many nuclei do I have, students? I have 2 nuclei. So 2 nuclei are present here. 2 nuclei. Students, how did we start the experiment or this entire mitosis? The entire mitosis started with 2 and 2C single nucleotide, one cell, and in the end, did we get the same 2 and 2C? Yes, same 2 and 2C with a single chromatid here, with a single chromatid. Yes or no? Yes. But we end up getting 2 cells. From one cell, we got 2 cells. from one cell, we end up dividing into two cells here. This was 2 and 2C. These two are also 2 and 2C. So students, can I call it equational division? Because there is no change in chromosome number from the starting to the end. In the middle, yes, there was in anaphase temporarily, but at the end of it, starting to the end, there's an equal number of chromosome. Diploid is maintained. So can I call mitosis, students? Can I call mitosis as equational division? Because there's no change in chromosome number, and that is the end of mitosis. It is called as equational division. It is called as equational division. Students, how many of you understood the beautiful process? This is happening inside you, all of you. It is happening all of you, inside you also, entire mitosis. So here's the NEET PYQ. Can I get the right quick answer? Here's the quick NEET PYQ. Can I get the answer, students? Can I get the answer? We'll just blaze through the entire this thing now. All of you. Spindle fiber attached to kinetochore of chromosomes during metaphase. Metaphase. Which of the following never occurs in mitosis? Never occurs in mitosis. Happens. This does not happen at all. This happens in the case of meiosis. This happens in the case of meiosis. Okay. Then select the incorrect statement with reference to mitosis. Incorrect statement with respect to mitosis. Splitting of centromere occurs at anaphase. Okay. All chromosomes lie at the equatorial plate. Yes. Spindle fiber attached to centromere. No. They attach to kinetochore. Not centromere. They attach to kinetochore. Okay. The growth of multicellular organisms due to mitosis. Obviously. Mitosis is called as equational division and it offers genetic stability. Yes. Makes sense. Makes sense. Right? That's why we grow. That's why we can grow, because of mitosis only. With respect to metaphase, which of the following statement is incorrect? Okay. Complete disintegration of nuclear envelope takes place. Obviously, complete condensation is happening. Chromosomes highly condensed. Metaphase are made up of, no, they're made up of 2 sister, not 4 sister. 2 sister chromatids. Wrong. Okay. And now, students, NEET 2017 question. 2017 question is right here. Anaphase promoting complex. Did we learn, students? Did we learn this today? Did we learn this today? Anaphase promoting complex is a protein degrading mechanical machinery necessary for proper mitosis of animal cell. If APC is defective in humans cell, which of the following expect to occur? Will not condense, will not be fragmented, will not segregate. Chromosome recombination of chromosome will not occur. Students, they will not segregate. That is, they will not go to anaphase only. They will not go to anaphase only, right? They will not go to anaphase itself. Condensation is happening. They will not segregate at all, because they will not go to the next stage. Because unless there is anaphase promoting complex, it is not going to anaphase only. Okay. Now, students, at the stage of mitosis shown in diagram, which stage is it and what it is? This is metaphase, first of all. Metaphase, right? So these two are gone. Spindle fiber attached to kinetochore, centromere splits. Centromere split happens in the case of your anaphase, gone. It happens. Option number 2 is the right answer, students. With that, we come to the end of today's class, students. End of today's class. Students, again, I'm telling you, students, question practice, test series are the most important. Whatever I teach you in my class, you'll get the notes, no worry. But what is more important is that, students, what is more important is that all of you need to do something called self-study. So after this video is over, I want all of you to watch it again. Doesn't matter, because this is a very important concept. Similarly, if you want me to teach meiosis, let me know in the comment section. Do you want me to teach the meiosis in detail, draw everything, show what happens in leptotin, zygotin, pachytin, diplotin, dikinesis? What exactly happens in all of them? If you want me to show diagrammatically, I will show it. Okay, students. So thank you so much, students. Bye bye. Take care, all of you. And meiosis, we'll do it next week. Meiosis we'll do next week. Do you want the same way meiosis also? I know. Because in meiosis also I can teach you the same way, very patiently, nothing hurry burry, all the time in the world. The more I hurry, more you will not understand. Okay, so if you want the concept clearing in for NEET examination, you should be in my class. Simple as that. Nowhere else. Okay, students. So thank you so much, students. Bye bye. Take care, all of you. If you're not a part of DNA batch still, if you're not a part of DNA batch, please, time is there still. There's the final call happening. If you're not a part of DNA batch, because in DNA batch you'll get everything, top to bottom, everything. I'll be teaching there. I am already teaching plant kingdom there. I finished cell cycle, cell unit of life, living world, biology classification. So I finished 4 chapters already there. I'm on my 5th chapter there already. Okay, and in DNA batch we'll be finishing the entire syllabus by December itself. Okay, and then we'll have 2 more revisions. Okay, students. So thank you so much, students. Bye bye. Take care, all of you, and I will see you in the next class. Until then, keep studying. Bye bye, all of you.